I'd like to invite our last speaker, Alex Rinehart from ViiV Manufacturers. Alex is the Senior Director of their Global HIV Prevention Strategy, and he leads the HIV prevention effort for ViiV. Welcome. Forwards, backwards, and then right.
Good morning, everyone. I'd like to thank the organizers, especially Gus and Rosalind, for the opportunity to give everyone here kind of a programmatic update on where we are with long-acting injectable Cabotegravir. But I'd also like to thank the three speakers before me, because a number of the items that I'm going to talk about have been nicely set up by each one of them. But I will be able to offer a little bit more detail on some of those items. So when we started this program, there were only two drugs that could be formulated as long-acting formulations. So Sylvain already mentioned rilpivirine, and then the other one was Cabotegravir at the time. And so Cabotegravir, I think everyone knows, is an investigational integrase inhibitor. It is being looked at for both HIV treatment and HIV prevention, but this morning I will only talk about the work that we're doing in HIV prevention. And what's unique about Cabotegravir is two inherent properties that it has. It has a very long terminal half-life, and it is highly insoluble. And both of those properties make it amenable to long-acting formulation. Sylvain nicely showed some of the data that were already presented a few years ago now in macaques, both with rectal and vaginal transmissions, but additional work has also been done with parenteral and penile challenges. And in all of those models, we've observed in macaques that Cabotegravir has shown a high degree of protective efficacy in those models. So I will not go into any of the non-human primate work anymore. So as I mentioned, Cabotegravir is being developed for two indications. One is for treatment, and that is a dual injectable regimen, so with injectable Cabotegravir and injectable rilpivirine. Rilpivirine was discontinued, as Sylvain mentioned, for PrEP, but was continued for treatment. But what I'm here to talk about today is the PrEP use for Cabotegravir, which is as a monotherapy agent, so the rilpivirine is not administered for PrEP. It is an injection that's being administered every 2 months now in the phase 3 trials, and I will come to those very shortly. So this is just a very simple graphic showing kind of the development program, both for treatment and for prevention. The green boxes, the green rows on top are the studies that have been conducted for treatment, for maintenance of viral suppression. But then the two at the bottom are the ones that I'm going to talk about. So there's a separate track for HIV prevention in MSM and transgendered women, and then a separate track as well for prevention of HIV acquisition in women. So this slide shows kind of the overall design that was used for the two phase 2 trials. And so in phase 2, you want to continue to demonstrate safety, but it's also an opportunity for you to make sure you have the right dose. So these are dose-ranging studies as well. And these, as was mentioned earlier, were done in low-risk individuals because these were placebo-controlled. And so the placebo injection for these two trials was saline. And so so there were two trials that were done. One was called ECLAIR. This was done in men at 10 sites in the United States. And the other trial was HPTN 077. HPTN stands for the HIV Prevention Trials Network. That's an NIH-funded research network. from the US. And this study included both men and women, and also went out to some more relevant areas outside of the US, including Malawi, South Africa, and Brazil. And to make a long story short, we started this program looking at an every 3-month injection. So that was two 2-milliliter injections. But the results from the ECLAIR study showed us that we were not achieving the plasma concentrations that we thought would be protective in humans with an every 3-month dose. So we moved to an every 8-week dose, which is what now has currently been selected for the phase 3 program. You will note out at the far right of this slide that originally, the primary endpoint for both of these trials was just looking, stopped at week 41, but given the results that we had seen from the ECLAIR study, we took the HPTN 077 study, which was in the field already, and actually extended follow-up to an additional 76 weeks, almost a year and a half after the final injection. And that's what's shown on this slide here. So these data were presented from HPTN 077 last year at the R4P, the Research for Prevention workshop in Madrid. And what you're looking at here is essentially the amount of drug that's remaining in plasma at certain time points after the participant's final injection. So 12, 24, 36, et cetera, all the way out to 76 weeks after their final injection. And if you focus on the orange bars, which indicate that the drug is no longer detectable in plasma, and you would expect over time that as you go further out from the last injection that the size of those bars would increase, and that's what we do see. But if you look at the top row, which are the results in men, you can see that almost all of the men had undetectable concentrations of Cabotegravir in plasma, with about 13% that still had low levels detected at a year and a half after the last injection. But as already alluded to, we see a different profile for women, and you can see that at 76 weeks after the last injection, there are 42% of women that still have detectable drug in plasma. There was another presentation at that same conference from R4P looking at the effects of gender or BMI on how quickly the drug is cleared from plasma. And so we'll start with BMI. So if you look at either gender moving left to right, so left are lower BMI individuals, and moving out to the right are individuals with higher BMIs. You can see individuals with higher BMIs take a longer period of time to clear the drug from their bodies, regardless of gender. But now if you compare genders, so if you're looking at the top row of men versus the bottom row of women, you can see that it takes almost twice as long to clear the drug from women as it does for men. However, what was also an important finding from this study is that none of these differences require any type of individualized dose adjustment by gender or BMI. So we were comforted to know that the dose that we have chosen for phase 3 is perfectly fine and usable regardless of gender or BMI. So this is just a summary of the phase 2 results. I've kind of gone through a lot of this already. What I've not shown, there's a large amount of patient-reported outcome data from both of those trials, 077 and the ECLAIR study, showing that the acceptability levels, either looking at injection pain, the acceptability levels were quite high with the injection compared to oral medication, and also the levels of satisfaction were very, very high as well. And that work is continuing on into phase 3. So you will have already seen this slide presented about the phase 3 design for the 2 studies that are in the field right now. So there's HPTN 083, which is the study looking at cisgendered men and transgendered women having sex with men, and then HPTN 084, which is the trial looking at adolescent girls and young women in 7 countries in Southern and Eastern Africa. So this is the common design across both of these trials, and I'd just like to take a second to kind of walk us through this. So you can see there are 3 different steps in these trials, and Paul has already referred to these trials as double-blind, double-dummy trials. And this is the unique challenge that we faced with, we're not comparing one pill to another pill here, we are comparing 2 different, wildly different modalities of administration, and it brings up many, many challenges about blinding and all of that. And so what you see here is a very rigorous design, a design that is rigorous to look at efficacy, but it doesn't really look at effectiveness. So this is work that will need to be continued, provided that we do see positive results for Cabotegravir. The Discover trial has already been alluded to here. So what you've seen this morning, HPTN 083, 084, and the Discover trials were the first of their kind to look at agents comparing against an active comparator. All the trials that had come before these trials, as Stacy had alluded to, were placebo-controlled trials, and she's already said and absolutely concur that that ship has sailed. We cannot do placebo-controlled trials anymore in this space. So in step 1, what you see here is what's called the oral lead-in phase. And so this was put into place years ago before we knew as much as we do now about Cabotegravir. And this is what we refer to as a safety check. So because you just saw how long the drug can stay in your body, what we wanted to do was put participants on oral therapy for 4 weeks to make sure there were no hypersensitivity reactions or any type of idiosyncratic event. And if there were, then we could pull them off of the oral medication right away before we put the first injection into them. I'm happy to report that all through the phase 3 program for both treatment and prevention, we've actually not noted any safety signals whatsoever, and we're actually, and I'll come to talk about this, in one of our treatment trials right now, we're actually moving away from the requirement of an oral lead-in period for use of the long-acting injectable. In fact, our intent, these are ongoing discussions with regulators, is to make it optional. So if you want to go through this oral safety check, you are certainly willing to, but if you want to do what we call direct to inject and skip over the oral part, then you are able to do that as well. The second step, so after everyone demonstrates good adherence in the oral phase in step 1, then everybody moves into step 2, which is the injection phase. And so as part of the double-blind, double-dummy design, everyone gets an injection every 8 weeks. Half of the people get active Cabotegravir, half of the people get a placebo, and then if you are on active Cabotegravir, then you're asked to take placebo Truvada every day, and if you are on active Truvada, then you take a placebo injection every 8 weeks. So this is the rigorous design that we've referred to. Obviously, we would never, ever do anything like this in the field. It would be entirely impractical. But this is the design that we were stuck with, knowing what we knew 4+ years ago when we started setting up the design of these trials. And then also another artifact of the design is step 3. And so these are event-driven trials, and I will come to that topic on the next slide. But at some point in time, these trials will end, and there will be a number of people who are still at risk for acquiring infection. And so after everyone leaves step 2 and moves into step 3, where you're no longer getting any injections, everyone will be put onto daily oral Truvada for 48 weeks to cover what we call the pharmacokinetic tail, which I will come to in a second as well. So this is actually an important slide, and it refers a lot to what Stacy talked about in her discussion. And so what you see here are all the locations of the sites for these 2 very large phase 3 trials. So all the orange flags are the sites for the MSM and transgendered women trial, and all the blue flags are the sites for the adolescent girls and young women trial. I will note that in South Africa and in Cape Town, there is an orange flag there. It's under the blue flag. So there is a site. It's actually a former IPREC site in Cape Town for the MSM and transgendered women study. I think it should also not be overlooked about what a monumental effort it has been to put these 2 trials into the field. Nearly 8,000 participants, which I will come to. It has been an amazing collaboration with the NIH, with the HPTN, with Gilead Sciences, to be able to put these, both of these trials into the field, and has really been a labor of love for all that have been involved. So there are some very unique considerations for the designs and conduct of these 2 trials. So the HPTN 083 study is being conducted in 4,500 MSM and transgendered women in the regions that you saw on the map, with a recruitment emphasis on some key populations, and I will provide some data in an update coming on that. And the HPTN 084 study is in 3,200 women between the ages of 18 and 45 in those 7 countries in Southern and Eastern Africa. As I mentioned, both of these trials are what we call endpoint-driven trials. So unlike treatment trials, which we've become all too familiar with, where we have endpoints where we see the level, what percent of people are undetectable at week 48, week 96, et cetera. Prevention trials are different. We have to wait for a certain number of events or seroconversions to occur, or a large enough imbalance between the 2 groups to decide whether or not an agent is superior or inferior or non-inferior, et cetera. And so, as already nicely outlined by Stacy, there is a very big difference between the conduct of these 2 trials. So because of the great results that we have seen in MSM with daily oral Truvada, it would have been almost impossible to try to conduct a superiority trial for any agent over an agent where we see levels of protective efficacy over greater than 90%. So what is the hypothesis that's being tested in the HPTN 083 trial is Cabotegravir at least as good as daily oral Truvada in this MSM transgendered women population? However, you saw the widely disparate results that we've seen in women in the sub-Saharan African setting, from VOICE, FemPrep, up to TDF2 and Partners Prep, a huge spread. And also in our discussions with the agency, they said there's no way that you can construct a non-inferiority margin given the results of these trials. Therefore, the women's study must be set up as a superiority trial, and indeed that is what the case is. All right, so here are the updates now, as of yesterday, for these 2 trials. So I am happy to report that as of Thursday night, the MSM transgendered women study is fully recruited. So we now have 4,507 men out of the 4,500 that were originally intended. That will be over-enrolled a little bit yet still with people who are in screening. What I think is even more remarkable is Sylvain referenced some of the enrollment goals that were set out at the beginning of this trial to make sure that we had proper representation of the communities that are most affected. And so the first was we wanted to have at least 50% of all the participants under the age of 30. And right now, we are at 66%, so almost 2/3 of the trial is under the age of 30. And then if you dig even a little bit deeper, and if you look at what the definition of an adolescent is for this trial's definition, those between the ages of 18 and 24, 40% of the trial are technically falling into the adolescent category. So I think these are numbers that are relatively unknown in this space, and I think there is going to be an absolute wealth of information and data coming out from these trials because of the populations that are being enrolled. Similarly, we set a goal to have at least 10% of the trial as transgendered women, and we are at 12% right now. And I will say, if you look by region, at the 5 sites that are in Asia, which are Vietnam and Thailand, 30% of their participants are transgendered women. I think it's 8% in the US, which is higher than what we had expected, and I think it's about 12% in Latin America. So already we have the largest trials ever conducted, ever enrolled for adolescents, youth, transgendered women, and now if we look at the special category of Black MSM, which is the epidemic that we face in the US, we set a goal for 50% Black MSM, and we have achieved that. So already we have the largest number of each of these populations enrolled just in one single trial. So I think that's really a remarkable achievement for the field. It says that it can be done. And so I think, we've been able to set a new bar here for being able to do that. And it's really due to the tireless efforts of the protocol team and all the special recruitment that was initiated for this. So I think this is really, really spectacular. Now, for the HPTN 084 trial, so this is the study in 18 to 45-year-old women in Southern and Eastern Africa. We are more than 3/4 enrolled now, and this study went into the field a year later than the MSM trial. So the MSM trial started in December of 2016. This study started in November of 2017. And I'm also happy to report and to say this also can be done. We are recruiting 200 women a month across these 20 sites. So we are looking at full recruitment of this trial sometime in mid to late spring of next year at a clip of 200 per month. And so the other one I think that is worth mentioning, if you look at this curve here, you will note that there is a very flat period between roughly May of last year and November of last year, and you might ask yourself, well, why is that? And so the May-June timeframe is when the issue of the potential signal for neural tube defects emerged for dolutegravir, and so that had some dramatic impacts on this trial, even though it was being conducted with cabotegravir. Cabotegravir is a sister molecule to dolutegravir, and it was decided that in the most taking into consideration the safety of the participants, that even though we had not seen anything like this in the cabotegravir program, there was a plan, a mandatory plan that was put into place that all women who were either already enrolled or about to be enrolled in HPTN 084 had to be placed on long-acting reversible contraception. So injectable Depo-Provera, NET-N, the etonogestrel implant, or an intrauterine device are the long-acting methods that are now in place. So during that 6-month period is when the women were reconsented. They had to go through all the regulatory approvals in country, but what the most important part is, look what happened after that. We were fearful that this would have blowback on the interest and enthusiasm for this trial, but you can see that we're bringing 200 a month into this trial, and so at least for the women in Africa, they did not seem to be bothered by this issue with dolutegravir, which is actually starting to become more mitigated as time goes on, and the data that were presented at IAS this summer. So there's some important sub-studies that are coming out of the women's study. So there is a pregnancy and neonatal study. So before we started HPTN 084, we believed that we were probably going to see 200 to 300 pregnancies based on data that had been conducted in the trials previously in that setting. But now, obviously, with the long-acting reversible contraceptive requirement, we're not going to even come close to that. So there have been a very low number of pregnancies that have occurred. And for those that do, they are shunted into this sub-study where we're going to look at cabotegravir plasma concentrations in each of the trimesters, cord blood, and breast milk as well. But this will largely be a commitment that will need to be fulfilled after the phase 3 trials are complete. Similarly, there's also a drug interaction study that will take place with the 3 long-acting methods that I mentioned, and then additional qualitative and quantitative work on patient-reported outcomes, tolerability, acceptability, preference, et cetera. There are also 2 important adolescent studies that are going to be conducted. These will start probably in first quarter of next year. These will be at select sites, 3 in the US for youth MSM, and then 3 at sites for adolescent girls and young women, 1 each in South Africa, Uganda, and Zimbabwe. So again, very important results that will be coming up. So we have already agreed with the regulators that all of the efficacy data will come from the adult trials. These are not designed as efficacy trials in the adolescents. These are designed as safety and tolerability trials to make sure that a younger population can tolerate these injections. So I think this is my last slide here. And so with innovation and novelty comes new challenges. So I've already mentioned the oral lead-in period. So we are working to have this made as optional, so that's already being taken care of. But then I've also referred to what we call the PK tails. So after you stop an injection, the drug remains in the body for some period of time, as I've already mentioned. And so this raises some unique questions about, well, what about the possibility for the development of resistance? We can talk about that in the question and answer period if you want to. It is a theoretical consideration at this point. We have, in all the animal work that we have done in which animals were infected, we did not observe any resistance in any of those models. But again, those are macaques. Those are not humans. And so we will hopefully learn about at least some of the answers for these questions from the phase 3 trials, and then also the concern about use in pregnancies. So, as I mentioned, out of safety, we did put this long-acting contraceptive requirement in place, but everyone wants to know, can the drug be used in pregnant women? And again, that's a question that will have to be saved for probably work after phase 3. So that concludes my part, and I look forward to the panel and answering any questions. Thank you.